Good afternoon, everyone, and thank you, Sunil and O2H. The goal and the mission statement of Ascend Bio is really to transform the efficiency and the economics of cell therapeutic development. And so, therefore, somewhat bold statement, it does in some ways represent the future of cell therapy. And to be very clear, we've got a unique intellectual property position, and the pre-seed funding has helped us file the PCT that we believe can drive the industry forward. Worth pointing out, when I say it changes the economics, the ownership of the intellectual property can really transform market access, the cost of developing these rather complex therapeutics, which of course have unique challenges. So that really is the intellectual and operational focus of this business. Few words on the opportunity. Clearly, we're going to try and generate curative medicines. So they're not bridge medicines. These are curative medicines for a range of major and orphan diseases based on the output of our technology platform. Few words on the opportunity. Cell therapy can revolutionize medicine. I think we know that with some of the advances that have been made in gene editing, cells, material science, where you can embed cells safely over extremely long periods of time. It's a massive and growing market, and I think one of my observations is in contrast to small molecule, it's very much partnership driven, and I suppose that's driven by the complexity of it, because you need the stem cells and you need to transform them into your desired cell type. You need to encapsulate, and of course, that constitutes the medicine. So it's multi-component by nature, which naturally lends itself to partnerships with big pharma, the well-funded Boston biotech, for example. And just as a point, clearly backed by the big VC companies, global biotech, global pharma, biotech, and a lot of drug discovery AI companies are investing heavily in this field, and they are potential partners for the company. So a few words on myself, along with Ludwig Valier, who's now grand title Einstein Professor of Regenerative Medicine at Berlin. Ludwig and myself founded a company, Cambridge enterprise company called Definigen, which generates, using CRISPR gene editing and stem cell technology, models for liver disease and diabetes. So that was our background. They sold quite heavily into the Boston biotech sector, and with some big pharma clients on top of that. But of course, the ultimate manifestation of stem cells is in producing curative therapeutics. So obviously there comes a point where it's time to move forward. This is, well, it may or may not be unusual, but myself, Ludwig, Cambridge University, and Massimo Tinzani, who's the head of liver down at UCL, we're the four shareholders in a company called Aculiv. And Aculiv was a cell therapy company with a liver technology, which we developed using our platform. And we developed that, founded the company, developed the asset, and sold it to a biotech, which is backed by Arch, Tencent, Milky Way, and Blue Yard. And it was quite a short operational period, because we delivered the asset. So, and that includes the selection of the stem cell, generating the liver cells with CYP450s, I think were mentioned historically. So that was the benchmark. They really had to have this high-end liver functionality, which is often lacking. And those of you who have worked in tox have probably got some scars from that. So that's really their performance improvement that enabled or incentivized this Arch-backed biotech to buy this company. And that happened, it was 9 months actually. Just within that, the work plan was, I think it was 1.5 years to schedule to deliver that asset. We actually got it done in 9 months, and that triggered the early acquisition. So just another minor point, I had a few roles at CN Bio actually, so it's probably the leading organ on a chip company. And that's important in this context, because what you're trying to do with cell therapy is encapsulate themselves and put them into the human body in a form where they're functional over the long term. And that's where this platform really does help you accelerate that phase of the asset development. Ludwig, he's still got his lab in Cambridge, but he was a professor at Cambridge, and he's recently moved to Berlin, where he's got a huge lab, couple of labs actually, and of course they're extremely keen to get involved in regenerative medicine. And he's done a lot of stuff, but I'd highlight two things. He set up effectively the stem cell platform for the MRC, which develops hundreds of stem cell lines and drives them mainly to different liver diseases. So he's got that background in terms of high throughput stem cell production and differentiation using the liver cells. He also set up a high throughput platform at Sanger, which the official title was Pancreatic Genetics. And again, that was taking donors, hundreds of cell lines, population relevant, and in a high throughput manner, generating pancreatic cells and generating disease models for diabetes within that. So that's our background, and it feeds into the company proposition. So this is the technology, and it really is a unique approach to the field. Effectively, Ludwig's responsible for a lot of the prior art in the field. A lot of it's gone to grant globally. And we've combined the two best-in-class stem cell generation technologies, which are direct differentiation and genetic forward reprogramming, the sort of synthetic biology approach. We've combined them into a single platform. That platform has a unique algorithm. And again, we use that engine to generate the liver cell type that led to that asset development. But that is a bridge therapy. So within this company, we've got completely new conditions, ownership of those conditions, and they will form the core for new therapeutic assets in the metabolic disease space. So how does this work? It's quite a grand claim that we can produce functional beta cells with a GCIS response, stimulatory index that's physiologically relevant, big claims. Why does this work? So this is what we did with Actualiv. We used preliminary outputs, so the technology's moved on a bit, but just it produced unprecedented set of conditions. Those conditions produced liver cells that express the CYP450s and the phase 2 enzymes and proteins. But crucially, it's that CYP450 activity that drove the functional cells, and that's what incentivized, and it's the differential, that fine detail in terms of cell functionality. Then partnered with a biotech backed by Arch and a lot of US and Asian funds, actually. I think Samsung were in there as well. But crucially, we developed the whole TPD, okay? So from the stem cell line, the cell generation in GMP compatible conditions, the encapsulation, the long-term POC studies, and the mouse study, which was probably the value inflection point. And so we're seeking to do that again with a lot of new assets. So in some respects, we're not doing anything new. We're just multiplexing, and we're going to put loads more zeros on these deals. So that's the play in the C fund. And again, just to reiterate, it was just within 9 months, but crucially, it was based on delivery. So it's not a theoretical, it's based on the delivery of that asset. And based on this company's press releases, that is proceeding into clinical trials. So this unique engine can produce cell types which will form the core of therapeutics in a range of areas. Metabolic disease, liver disease, respiratory, gastroenterology, and lots of orphan indications. So where we are now at this pre-seed stage, we've generated conditions for pancreatic cell types. I wasn't sure about the confidentiality in the audience.
No confidentiality. It's been zoomed around the world.
Yeah, no.
No chat.
So what I can tell you in relation to the pancreas, as at this stage, we have a TTP and a pancreatic cell asset. Crucially, it uses a hypoimmune HLA optimized stem cell line, which has a drug master file associated with it. Okay, and that's crucially important for these sort of therapeutics, because obviously that's a huge play in the field. Just on a slightly wider point, and it's really just this year, I'd say that a lot of players in the game are coming through with whatever you want to call it, immune optimized, hyperimmune stem cell lines that they believe deal with the rejection issue. And again, I reference the high throughput platforms that Ludwig's been involved in. And so all our technologies are designed to use a range of donors. So for example, historically, we've used around 20 FDA compliant stem cell lines, and they all work with the platform. That's hugely important because if we're going to partner with huge players in the field, there's an extremely high probability that, let's say, their hyperimmune stem cell line will work with our platform and produce pancreatic cells, lung cells, etc. So that's the play, and I think that's a really important point to get across. You know, we've always structured these technologies in a way where we can use lots of FDA compliant stem cell lines. So, Ascend Bio, company's been set up to fully exploit the platform, clearly focus on some big plays, pancreas, lung, liver. Ludwig's a member of the Cambridge Cystic Fibrosis Hub. I think there's 3 key streams, and he's one of them, involved in the cell generation fairly predictably. And of course, you know, you can see that we're involved in all these big plays, but clearly there are, particularly around Boston, you know, working within the Orphan Drug Act, there are lots of niche therapeutics that Boston biotech and the VC community are investing heavily in. So we've got an interest there. If you're interested, I can guide you through the technical background, but crucially, when you combine these two market leading approaches, you get elevated function. So very simplistically, that's the play. You elevate the function by using this sort of integrated synthetic biology approach to use a developmental wave to produce the high-end function. This is the most anemic slide, apologies for that. But for example, what I did want to reference is the core of the business is therapeutic assets, okay, because that's where the money is, we believe. But for example, Ludwig is on the Open Targets Consortium with a range of liver pharma companies, and I can't give away the confidentiality, but he has produced new NAFL targets, and they need to be validated. And so we've got a platform to do that. So in the service offering that we'll take out to pharma, it's structured on that. So there's a follow through from our sort of founding expertise there. Deals in the space, you know, how do you get return on your investment? These are just some deals, big numbers around diabetes, as you'll know. Doug Melton is a Harvard professor. He was the big name. So although his company was preclinical, you know, he'd had probably 50 or 100 million of funding that ultimately went into that vehicle and was bought by Vertex. Vertex also bought ViaCyte, which was, it was a trailblazer, but it was kind of running out of money, I think, for a paltry 320 million. More recently, Takeda made a strategic investment in a stem cell company called Tigenex, and then they, as it went through the phase trials, they purchased the company. Closer to home, Mogrify did a deal with Astellas, who made a strategic investment, and that was to generate sets of transcription factors to develop a cell type. Clearly, we have that capability within the organization. Again, close to home, BitBio did a deal with BlueRock, which is they had a press release out about that, to identify conditions that can generate Treg cells. We've got capabilities to do this with our engine in the 118 that we referenced in that previous slide. So you can see how we can work with pharma and biotech, lots of different, I would call them tiered ways in the therapeutic space and more the wider drug platform development space. So what's the ask? Probably a moderately large seed investment. So we're pre-seed at the moment, grateful for the investment from O2H, seeking 5.5 million, really to repeat what we did with Aculiv. So we're not going beyond our sphere of competency. It's just to progress liver, panc, thymus, and lung. So duplicated something we've actually already done. I would point out that we'll also seek to develop a lot of high-value partnerships because of this cell generation capability. In terms of the financial model, in the 2-year seed phase, there's no cash sensitivity, so we're pretty comfortable generating these assets. And then there would be a stage gate analysis. So I've got a financial model which really, and operational model, which details the costs to continue internal development at the Series A and take it to end of phase one, be around 23 to 30 million. And because obviously want to have the option, if it's a really good play, competitive landscape, then we want to continue the internal development. So the Series A would be anything between 8 and 30 million. So I'm going to be dragged off. So I'd like to thank you for your time, and any questions you have, happy to take them.